Our next oration, Dr. S.C. Lahiri Oration, is a prestigious academic award established in honor of Dr. S.C. Lahiri, a pioneering Indian pharmacologist. This oration is conferred on distinguished researchers who have made significant contributions to pharmacology and related biomedical sciences. By recognizing scientific excellence, it commemorates Dr. Lahiri's legacy and promotes continued advancement in pharmacological research and education. For chairing this session, we have Professor Sharma already with us. I would like to invite Dr. Sukhbir Kaur to join him on the dais. Your presentation, okay. So, now I request our session chairs to introduce our orator.
Hello. Good afternoon, everyone. And thanks to the organizers for giving me this opportunity to chair this session. So the speaker for this oration is Professor Sadhana Sathaya. She is a senior professor of pharmacy at the Institute of Chemical Technology, Mumbai. She holds a PhD in pharmacology, M.Pharma in clinical pharmacology from Grant Medical College, 1986, and B. Pharmacy in pharmaceutical sciences. Her research focuses on the development of herbal-based therapeutics for CNS disorders, pharmacokinetic pharmacodynamic modeling, metabolic diseases, immunomodulation, and phytochemical anti-cancer approaches. Professor Sathaya has supervised over 100 postgraduate and doctoral students and collaborated with leading institutions such as King's College London and IIT Bombay. A fellow of Maharashtra Academy of Sciences, she is a recipient of DST Vidushi Fellowship and multiple Best Teacher Awards. With this brief introduction, I welcome you, ma'am, to please give your presentation.
Thank you, everyone, and good afternoon. I would like to thank the organizers for inviting me to give this presentation. My title of my presentation is From Blood to Brain, a translational strategy for early diagnosis and targeted therapy in Parkinson's disease. So, I would like to dedicate this presentation to Professor S.C. Lahiri, who is a renowned neuropharmacologist who worked in the area of venom, immunology, etc. So, about Parkinson's disease, if you know about it, that it's a really frustrating disease wherein you find many movement disorders which make your life difficult. So, current status of this neurodegenerative disorder, wherein there are neuropathological features. You can see that there is an alpha-synuclein, which is a protein, otherwise good protein, which helps in synaptic transmission, that gets misfolded. Normal monomeric form gets misfolded into beta pleated sheets, and that starts precipitating out, and that produces neurodegeneration and neuronal loss. So because it gets precipitated out, the microglia and the astrocytes, they get activated, and there is a blood-brain barrier leakage and neuroinflammation. And if one cell dies, when there is a one neuronal cell death, particularly in the area of SNPC, substantia nigra, many other cells also progressively degenerate, and thus the disease is progressive, complex, and multifactorial, particularly affecting the body movements, simply because dopamine controls the different body movements. As far as clinical diagnosis is concerned, the diagnosis is mainly based on symptomatic, meaning symptoms which appear, and 70% neurodegeneration when it appears, the symptoms start appearing. So, it's very difficult to diagnose this particular disease at an early stage, diagnosed only when 70% neurodegeneration takes place, particularly by all these invasive technologies, SPECT, PET, DOPA-PET, CT, etc. So as of today, there is no reliable, easily applicable and cost-effective diagnosis, and of course there is no cure. So it's the need of the hour that we diagnose this disease at an early stage so that we can intervene, we can have disease modifying agents for early intervention to manage this disease and give a good quality of life to patients. Now, what do the stalwarts they say? Professor Ole Isaacson from Harvard Medical School, he says that blood-based alpha-synuclein signatures could revolutionize the diagnosis of Parkinson. And Professor William Langston from Stanford University and CEO of Parkinson's Institute, he says that it is the only unifying thread in Parkinson's, which this is a target that holds a promise of true disease modification. So if you identify it early, you can give disease modifying agents in this case. So what is the research overview? So we produce polyclonal antibodies in rabbits with a high titer after each successive immunization, a high titer of purified antibodies specific to beta cross sheets, meaning the misfolded alpha-synuclein were achieved, and the antibody was further tagged with fluorophore, which could be optimized later in a NC-FLISA assay. The antibody specificity to the misfolded protein only and not to the monomeric form, because monomeric form is good, that was validated, and then the detection of alpha-synuclein protein, misfolded protein using NC-FLISA was done in vitro and in vivo. So this was the proof of concept, like we wanted to do that so that we could go ahead. Now I'm just going ahead with this slide because I'm showing you in this. So this was confirming the selectivity of polyclonal antibody to beta cross-sheet form. So this is the beta, the green ones are beta cross-sheet form and the red ones are monomeric, that is good form. So we validated that our antibody only bound to the beta cross-sheets and not to the alpha cross-sheets, alpha monomeric. So that means that we have validated that it only binds to the beta pleated sheets, and see the sensitivity, it's from 1 picogram to 1000 nanograms. So you can say that 1 microgram per ml also could be detected, and the least that we could detect is 1 picogram. And if you see the blood levels of misfolded alpha-synuclein in the Parkinson's patients, it is about 30 to 50 picograms per ml. So we are quite in a we are good as far as our method is concerned. Now, okay, now this was direct. Now, when there is a misfolded synuclein in the blood sample, we have to use this competitive FLISA method. So this competitive FLISA method is particularly between the interaction of the misfolded protein in the sample, biological sample, whatever it is, and the one which is coated on the nitrocellulose membrane. And so it means that the increase in the concentration of the blood of misfolded protein in the blood serum will definitely show decreased fluorescence in the coated antigen. So this is what it confirmed. This is the fluorescence versus biological sample concentration of alpha-synuclein, and we could detect it, meaning in this competitive method. This is a little different method than the first one that I showed. It shows again very good sensitivity between 1 picogram to not 100, also 1000 picogram per ml, and the concentration in the blood sample of a Parkinson's patient which is being reported is 30 to 50 picograms per ml. Now we had to confirm it using in vitro and in vivo method. So in vitro method was sushi cells, wherein we had added rotenone, a known pesticide. It's a well-known model to create Parkinson's in animals. So we saw if it could produce the same markers in the sushi cells, and we saw that control shows highest fluorescence, meaning less misfolded protein, and at 24 hours after rotenone addition, the fluorescence reduces. At 48 hours, it is not detected at all. So this proves that you can detect the misfolded protein concentration in the cell supernatant, and similarly in the rat serum. The rat's brain was injected with either rotenone or misfolded beta sheets of alpha-synuclein. It was directly administered or injected into the SNPC area, and at different times, 7 days, 14 days, till 6 weeks, 42 days, we kept on taking the blood samples just to understand whether we are able to detect the concentrations of misfolded beta pleated sheets. And you can see that the sham control, where nothing was administered, it shows good fluorescence, meaning no misfolding. Whereas at 7 days, both rotenone as well as alpha-synuclein, it showed fluorescence, meaning, and a little less fluorescence than the sham control, meaning the protein has started misfolding because you have added something into the rat's brain area, and then slowly over the time, there is absolutely no detection of fluorescence, meaning the increase in the beta folded sheets is there. So this also proves the concept that we concentration of the beta cross sheets increase in the reduction of fluorescence intensity, and the proof is proved. The POC is done both in vivo and in vitro samples. Now, okay, this was through the blood samples. Now, can we do IHC with the polyclonal antibody that we have produced? So we took the SNPC areas of the administered alpha-synuclein rat's brain and rotenone induced and sham control. You can see that beta cross sheet induced rat brains show higher fluorescence. Again, this is direct method. So you are just detecting the fluorescence. Here, higher fluorescence meaning higher misfolding, and you can see that beta cross sheet, it gradually increases. Even rotenone gradually increases, and sham control does not show any fluorescence, meaning where you have not added any misfolded beta sheets, you don't see any fluorescence, meaning no misfolding. So these results are just put here. This is confirming that not only from biological samples by competitive NC-FLISA method, you can even use this antibody to detect the misfolded alpha-synuclein in the brain samples as such for immunohistochemistry. Now, okay, so we understood that you can diagnose the disease at an early stage. You can diagnose as low as 1 picogram per ml of misfolded protein in the rat's blood sample for that matter. Then let us see what early intervention that we can do. So currently available treatments, if you see, it's only dopamine producing agents. Either it is dopa decarboxylase inhibitors like carbidopa, or it is COMT inhibitors like entacapone, or it is MAO inhibitors like selagelin, or something like amantadine that directly increases the dopamine concentration, or it is dopamine receptor agonist. So whatever it is the increase in the dopamine levels, and the relief is only symptomatic. And you all know, maybe if you have seen Parkinson's patients, those who are on this syndopa or dopamine kind of treatment, they always will have dyskinesia. That's the major side effect because of oxidation of dopamine in the body. So what next? So all that we need for health and healing has been provided by God in nature. That's what even Madam Gulati told us. So this is the same thing, all the challenge is only to find it. So we were working on this antioxidant luteolin. It's not lutein, it's luteolin. It's a little different. And you get it in many of the vegetables. It's a flavonoid. And we have worked on it on the Eclipta alba for treatment of epilepsy. And in the market, I have seen it in the US market, not in Indian market. It is a nutritional supplement for cognitive health. So generally people take it for antioxidant effect of it. I was interested in its anti-inflammatory and neuroprotective property, and mechanism is suppression of NF-kappa B and TNF-alpha lowering, and it has, because of its antioxidant properties and modulation of microglial activation, it produces neuroprotection as well. If you see the pharmacokinetics, although half-life is 5 to 7 hours and appears in the bloodstream in 1 hour, we have found that it is extremely lipid soluble. It's not water soluble at all, and it has got a very low permeability and brain biodistribution particularly. Now this is how the pure luteolin looks like, and this is what the formulation that we have developed in ICT looks like. We made it with help of a pharmaceutical technologist, and we are calling it Neura Health because it has shown good effect in vitro PD model in sushi cells and in rat model of PD. Now let us check what are the changes that occur in the brain. So if you see the normal brain, this is BDNF, which protects the brain, which produces healthy neurons. This is tyrosine hydroxylase, which is a rate limiting enzyme to produce dopamine in the body. This is tripeptide glutathione, which is an antioxidant, and these are two antioxidant enzymes which provide antioxidant effect. And this is KEAP1, it's holding on to another factor that's called NRF. So the levels of KEAP1 are low, NRF is good, antioxidant effect is good. And when you have Parkinsonism, when the alpha synuclein starts misfolding and you have Lewy bodies, then there is a neurodegeneration in the SNPC area. And because of that, the KEAP1 increases, the GSH SOD catalase will go down, the glial fibrillary microglia get activated. Because of this, you will have high inflammation and neurodegeneration as seen by the lipid peroxidase, high lipid peroxidase activity. So when this happens, the mitochondria get damaged. They are no more healthy, producing healthy movements. They are now damaged, degenerating the dopaminergic neurons and producing uncontrolled movements. So what? So we can determine if the Neura Health, which we have produced, is it better than luteolin alone, API? And these were the certain markers that we used. So I'll show you directly the results. So these were the results of in vivo efficacy of luteolin. So here we were just looking at the API, which showed antioxidant properties, reduced reactive oxygen stress, reduced KEAP1 expression, meaning increased NRF1, NRF2 and GSH SOD catalase. LPO was reduced, indicating that neurodegeneration was reduced, cellular membranes were stabilized, GFAP levels were reduced, and we saw higher TH activity, tyrosine hydroxylase activity, so neurogenesis, and we saw less misfolded alpha synuclein. So this was done by using our antibody. So neuroprotective, anti-inflammatory generation of dopamine producing neuron. So everything that would help in modifying the disease. So we identified that we should work on this particular luteolin and make it more available to body. So we developed it for better bioavailability and brain biodistribution, and solubilization was achieved with using selected excipients, of course with the pharmaceutical technologists, Sutix people, and this is how the formulation looks like. We have filed a patent on this, which is also published, and we are awaiting the results now, from the patent office. Now, we have to see whether whatever that we have made is really available in the body, whether it increases permeability. So if you see the API release versus the Neura Health release in SGF and SIF, you can see it is 3 times higher release. 3 times higher release of the formulation in vitro methods in SGF and SIF. And in vivo PK studies indicated the Cmax was increased 1.5 times, the brain distribution was increased 3.5 times, and the Tmax came down by 1/6, meaning 36 hours to, this is what we found in rat. And now we wanted to see if along with this, whether it correlates with improved grip strength or motor coordination or locomotor activity, because that is what is affected in the Parkinson's disease. So whatever results that you get in PK, higher brain distribution, whether it really translates into pharmacodynamic activity. So we chose a model of Parkinson wherein we added rotenone into the brain of SNPC area of the rats, and these were the different behavioral parameters using rotarod, muscle strength and coordination, open field model for locomotor, mitochondrial health, complex 1 activity, GSH, all these from brain homogenates for antioxidant, and LPO from brain for neuroinflammatory. And these were the IHC markers, so BDNF, GFAP and tyrosine hydroxylase. This is what we saw from IHC. So, you know, whether the increased motor coordination correlates with antioxidant activity and neuroprotective activity as we visualize, and does the IHC confirm it? It's all what we wanted to see. So these are the results. I've just put it. You can see that rotarod, you can see 15% improvement by Neura Health, 30% improvement in locomotor activity, 60% higher improvement in complex 1, so more energy, better mitochondria, 85% of glutathione, increase in glutathione, which is a natural antioxidant tripeptide in the body, increase 37% and 54% of SOD and catalase, and 35% lower value of lipid peroxidase, indicating less cellular damage. You can see that BDNF increased 20%, which helps in normal healthy growth of neurons, and dopamine producing neurons 36% increase, and you can see that inflammation was reduced by 10%. So I can now say that it exhibits 10 to 85% better neuroprotective or anti-PD activity than the plain luteolin, meaning whatever that excipients we had added for better solubilization and brain biodistribution has worked here as seen by in vivo rat model. So the good news stories in medicine are early detection and early intervention. So yes, if we could detect the disease at an early stage, say 15 years early, and you can, give a patient. I'm not saying that the patient has to suffer, I mean, good if he doesn't suffer, but he can take, if he finds a higher level of misfolded alpha-synuclein, he can take this luteolin for that matter. And so what is that translational strategy is that we have developed a novel polyclonal antibody. It was polyclonal, it's not monoclonal. With a simple setup at ICT, we have produced it, we have purified it by simple methodologies. We have confirmed its specificity only to the misfolded alpha-synuclein, and in blood samples also we could detect it in rats' serum. And so it's a non-invasive, few microliters of blood sample, serum sample that we require to analyze this antibody. And then optimizing luteolin formulation for better solubility, bioavailability and brain biodistribution. So all that it did is, of course we had kept a group of rats when we induced rotenone into the animal brain. There was a group where we had also started giving them luteolin formulation. So it improved motor coordination, attenuated oxidative stress and neuroinflammation, I just showed you in the previous slide, and dopaminergic neurogenesis. So early diagnosis and early intervention resulted into early neuroprotection. So this is a similar slide. I just want to tell you that the range of PD patients is between 30 picograms and 50 picograms per ml, and we could detect it at 1 picogram to 1 microgram, so wider range of detection. And yes, luteolin, Neura Health showed better results than API. Now looking at the market potential, it's a easy and scalable technology, very easy and scalable technology, with significant higher solubility and brain biodistribution, confirmed anti-Parkinson's and activity in preclinical cellular and animal models. And you can see the global market, almost 1900 billion US dollars market, which is expected to grow at 7.7% till 2033. So there's a huge market potential for this, and I'm so proud that recently I received a Vidushi grant from DST for early detection. So, whatever proof of concept that we have done in the animals and in cellular studies, we've got money to study it in the clinical setup. So we have already connected and collaborated with biggest municipal hospitals in Mumbai, and we are taking it ahead to at least understand if the diagnosed clinical patients of Parkinson's do show certain levels of misfolded alpha-synuclein as against the healthy people for that matter. And of course, if the luteolin that we have made, that would take a little more time, but I'm going to use the normal luteolin that you get it in the market as a nutritional supplement. If that is given, is there any difference? So that's what we are going to see with this funds that we have got. So thank you so much, and I'm happy that I finished it in time. And this is my team. She's Paramita, who actually developed, tagged, and studied this polyclonal antibody, confirmed everything. So she is Paramita, who is now working in Iceland in Alvotech Biotech Company. And Priyanka was an MPharm student who worked on luteolin formulation and its confirmation in vitro and in vivo models. She is now currently doing PhD in America, St. John's University. So thank you very much. I'm open to all questions.
Yeah. Great. I really appreciate Professor Sadhana for your excellent talk, actually, and excellent oration talk. I think you have really gave the idea from like bench to bedside. So that's one of the things actually, like translational point actually you have taken into consideration, and I've seen that you have done in vitro studies, then in vivo, and then going into the human with history with luteolin formulation with NeuroHealth alone and their combination. I think excellent work. I think we must give a big hand.
So, in the morning there was a speaker. She was a clinical pharmacologist, and she said, what's the use if you, like study so many things and it just remains in your thesis? So you have to see to it that it sees the light and we are able to help patients in some way or the other. So that was the motto. Thank you. Any questions? I'm open. Yeah.
Yeah, sorry. Actually, I think in oration session, you can interact with actually the speaker after this session, but generally, like we are not having question answers.
Thank you.
Yeah.
We will now felicitate our orator.